Okay, so may I start?
Yeah, sure.
Thank you. So I would like to start by acknowledging the online summit organization for the invitation and also to be able to participate in this summit on food science and nutrition. Today, I will be presenting a part of a broader research project entitled Temperature Effect on Listeria monocytogenes biofilm, um, and we assessed virulence using phenotypic and genotypic characterization of the isolates that were collected in ready-to-eat food industry. Listeria monocytogenes can lead to listeriosis, which is a severe illness almost exclusively transmitted through food consumption. Food products contamination occurs during food processing, in any step of food processing, and there has been a significant increasing trend in listeriosis in Europe with high fatality rates. This is the highest rate of any foodborne agent. And in risk groups, the fatality rate peaks up at 20%, which is quite high. Listeria monocytogenes is a very important pathogen or opportunistic pathogen, and it assumes a great importance in public health since it is a cytotropic opportunistic pathogen. It has the capacity to proliferate in a large variety of food-related environments, and also food matrices. Moreover, Listeria monocytogenes is able to produce biofilms, which is the predominant form of bacteria in the environment. Biofilms are associated with serious hygiene problems because they exhibit a higher resistance to sanitizers than the planktonic forms of the bacteria. So, disinfection of food contact equipment is a critical step for food producers. However, developed resistance to biocides has been described for Listeria monocytogenes, which is a topic of concern in the context of food safety and public health. So in this study, Listeria monocytogenes isolate from food and food-related environment samples were collected in the ready-to-eat food industry, and these isolates were assessed using genetic and pulsed gel electrophoresis typing to reveal potential sources and routes of contamination. Also, virulence was studied through biofilm forming ability testing at different temperatures, as we will see ahead, combined with the biocide susceptibility assay to further assist the food industry, in delineating and establishing preventative measures. To attain those aims, Listeria monocytogenes isolates were collected during 6 years from food and food-related or food contact surfaces from a ready-to-eat food industry located in Alentejo, Portugal, for food safety verification purposes. Species-specific identification and serogrouping were performed using a multiplex PCR according to Kedwaton, and PFG typing was done according to the PUSNAD standardized procedure for Listeria monocytogenes. Selected strains, together with four reference strains, were used to have representatives from different serogroups and different pulsatile for the biofilm and biocide assay. Biocide activity testing was performed using an European standard for bactericidal activity evaluation of chemical disinfectants used in food and food industrial areas. Commonly used biocides in those premises were evaluated, and so we selected an oxidizing compound, an ethanol-based compound, a quaternarial ammonium compound, and also benzalkonium chloride. In this slide, we can see a summary of the statistical models and software we use for data analysis in this work. On to results, 26% of the isolates were confirmed as Listeria monocytogenes, and they belong to serogroups 2A, 2B, 2C, and 4B that are altogether responsible for more than 90% of human listeriosis. Postal gel electrophoresis typing revealed that pulsotypes were grouped in 6 PFG types, and we even gathered 3 clusters in this analysis, in this pulsotype analysis. The first and the biggest cluster has more than 50% of the strains, all belonging to serogroup 2C, and these presented identical restriction patterns indicating a possible common source. The next cluster, the middle one in this slide, has serogroup 2A strains that were collected from different finished products, and we can see that pulsotypes FC7 and FC9 that were collected with 1 year of interval presented the same pulsotype, which is suggestive of persistence over time within the food premises. The last and smallest cluster in this slide included strains that were collected during 2 years, 2 consecutive years from different raw materials. Because pulsotypes that were identified in raw materials exhibited a high similarity with those from equipment and also finished products, we think this suggests a repeated reintroduction of strains into the food unit over time. Um, Considering biofilm, the biofilm assay selected strains representing different serogroups and pulsotypes were selected for this assay. We performed an in vitro biofilm assay at 30 degrees, 20 degrees, and 12 degrees. All the strains we selected were weak biofilm formers, and crystal violet particle density revealed significant differences in the biofilm forming ability, whereas using viable cell counts, no significant differences were detected. Serogroups and biofilm formation revealed no significant differences. For other biofilm forming ability testing at 20 and 12, we further selected 5 strains based on serogroup and their biofilm forming ability parameters at 30 degrees. Although there were no significant differences from biofilm formation at different temperatures, crystal violet particle density values presented significant differences. So at 30, we could see that the highest biofilm formation occurs. As for the biocide activity testing assay, biofilms, our biofilms were susceptible to the oxidizing compound, to the ethanol-based compound, and also to benzalkonium chloride within the tested concentration range with 5 minutes of contact. But they were not, for the biofilms we developed in vitro, they were not susceptible to quaternary ammonium compounds, even when exposed to 150 parts per million. Some strains presented less than 50% of reduction. So, quaternary ammonium compound was needed in higher concentrations than the ones that are commercially recommended by these biocides manufacturers. LD90 values reveal that the higher the biofilm formation, the higher the LD90 values for quaternary ammonium compound and also for benzalkonium chloride. Taken together, our results emphasize that Listeria monocytogenes virulence in food-related environments must be carefully addressed. The strict selection of suppliers and raw materials to prevent repeated introduction of strains into the food facility should be considered. Also, the cautious use of sanitizers should be considered as well, as biofilms might select strains that are adapted to the food environment and that are resistant to some of the sanitizers that are used in those premises. Our results with quaternary ammonium compounds and the resistant strains underline the need for these industries to use sanitizers in rotation schemes. Sorry. Finally, I would like to acknowledge the research project mentioned in this slide and also the collaborating institutions. Thank you so much, and I am now at your disposal for any questions or doubts that might have um, uh, that you might have. Thank you.